From Module 1, you learn that scientists are interested in studying gene products for added insight into cellular function. RT-qPCR is a highly effective and popular method that is currently used in many fields of scientific research. This method allows scientists to get a closer look at how certain variables affect the synthesis of gene products. Gene products, such as proteins, determine the function and therefore the purpose that a cell carries out. These gene products are not static but fluid in their makeup depending on environmental cues. To better orient RT-qPCR regarding how scientists study gene products in an experimental capacity, let's first review the basic scientific dogma of biology. The current slide illustrates how a cell interacts with its environment to adapt and survive. Starting on the left-hand side of the slide, we recognize the familiar packaging of genes within the cell's nucleus. During its life cycle, cells will receive signals from the microenvironment that induce transcription of genes. Transcription, a process that we will uncover in more detail later, is the process by which a cell synthesizes different transcripts from genes. These transcripts comprise important information that are used by the cell as a form of transcriptional and translational regulation, or they can be used to transmit messages in a process of protein synthesis called translation. At any given time during a cell's life cycle, it will contain a milieu of different transcripts that make up the transcriptome. The transcriptome can contain information that uncovers more about how the cell is functioning, which is often important for scientists to infer the pathology of cells. RT-qPCR is a method that aims at identifying targets within the transcriptome to ask questions like, is the particular transcript present? And if so, how much of it is present in comparison to other targets or other cells of interest? To learn more about what targets might be of interest to a scientist, let's explore the general makeup of a cell's transcriptome. In the mammalian cell, incredibly 80% of its genome is actively transcribed into the transcriptome. This statistic is powerful in its suggestion that much more of the DNA that was originally termed as junk may actually play a far more important role. Within this large library of transcripts, roughly 90% is assigned to the general RNA biotype termed non-coding RNA. As the name suggests, non-coding RNA refers to RNA transcripts that do not code for proteins, yet play pivotal roles in mediating transcription and translation. Some common non-coding RNA biotypes include small nuclear RNAs, short hairpin RNAs, transfer RNAs, and ribosomal RNAs. Ribosomal RNAs make up the majority of transcripts within the transcriptome due to their large functional requirement during translation. Because of their overwhelmingly and steady presence within a cell, quantifying ribosomal RNAs is used as a positive control during RNA extraction. However, after RNA isolation, scientists utilizing RT-qPCR may utilize a ribosomal RNA clearance step to reduce their inherent noise and enrich the signal from other RNA transcripts of interest. The other well-recognized class of RNA biotypes is termed coding RNA. Coding RNA amounts to a humble 2 to 5% of the transcriptome and describes transcripts that contain a code or message used during protein synthesis. Therefore, these transcripts are also collectively known as messenger RNA or mRNA. Although mRNA is vastly underrepresented within the transcriptome, it is most often targeted in RT-qPCR experiments due to its vital importance to predicting the proteome makeup and therefore the current function of the cell. In the context of gene expression studies within the RT-qPCR world, scientists are generally interested in capturing the protein synthesis potential by looking at the mRNA of a cell. To better understand the link between a gene, mRNA, and protein, let's explore the process of transcription and translation a little closer. During transcription, DNA is unpacked to promote access of transcription machinery to one or more genes. Genes are made up of two complementary strands of DNA. The complementary strands are paired by hydrogen bonds between two nucleic acids. Each thymine pairs with adenosine, and each cytosine pairs with guanine. Genes are unwound during transcription to expose the complementary strand that is used to create the new transcript. RNA polymerase and associated transcription factors recognize the unwound gene, whereby RNA polymerase facilitates in the pairing of complementary nucleic acids to form an RNA copy of the gene sequence. During transcription, RNA polymerase incorporates uracil rather than thymine into growing transcripts. Thus, the RNA copy of the gene will contain a slightly different combination of nucleic acids than the DNA. The newly synthesized single-stranded transcript will undergo processing that leads to the addition of a 5' guanine cap and a 3' polyadenosine tail. These processing steps enable stabilization and protection of the mature mRNA molecule while it awaits its fate within the cell. As stated before, non-coding RNA transcripts can play pivotal roles in regulating steps within transcription and translation. The mature mRNA transcripts will eventually be translated into proteins through the complex process of translation. Translation involves both ribosomal RNA and transfer RNA to facilitate the ribosome in efficient protein synthesis. When learning how RT-qPCR is utilized to study gene expression, reviewing these fundamental principles will prove substantial when we begin exploring considerations for sample preparation and the initial RT-qPCR protocol. Let's review some of the characteristics of RNA before continuing in on these topics surrounding RNA isolation. To review and expand on some of the key characteristics of RNA biotypes, I've created a short list below to reveal some of the main similarities and differences between coding RNA, or mRNA, and non-coding RNA. Let's start by examining mRNA first. As you already know, mature mRNA transcripts are characterized by a coding sequence or message. This message is what contains the information utilized during translation. The 5' end of mRNA is equipped with a guanine cap, and the 3' end has the addition of a poly-A tail. These additions provide a bit more stability to mRNA while it continues towards translation within the cell. However, even with these stability buffers, mRNA contains a very short half-life of minutes to days, which is why scientists are extra careful when handling it in the lab, something that we'll touch on later. Now let's examine a few characteristics about non-coding RNA transcripts. We learned earlier that non-coding RNA encompasses a diverse group of RNA transcripts that do not contain a coding region. These biotypes range in size from 19 nucleotides all the way up to 100 kilonucleotides. Although a few non-coding RNA classes contain modified sequences, most do not contain the 5' or 3' processing caps that mRNA contain. Similar to mRNA, non-coding RNAs also represent an extremely unstable group of molecules, so extra precautions are taken when handling these in the laboratory as well. Now that we've reviewed the processes involved in gene expression and identified the diversity and characteristics of RNA biotypes, let's dig into the basic workflow steps involved in gene expression analysis. The current figure outlines the key steps in gene expression analysis utilizing RT-qPCR. For the duration of this module, we'll be focusing on steps 1 through 2, the isolation of the RNA and the synthesis of complementary DNA, or cDNA. To visualize these first two steps, let's imagine a scientist attempting to investigate a cell line for gene X expression levels. The sample of the cells would first be used to harvest or isolate the desired collection of RNA biotypes. Then, the scientist would use the RNA to generate cDNA for downstream amplification steps. Before getting any further, let's pause and examine step 1, isolating RNA. Extracting a pure RNA sample is a crucial first step when achieving success when utilizing RT-qPCR for gene expression analysis. The nucleic acids extracted from biological material need to be sufficiently free of components which may inhibit enzymatic reactions. Semi-quantitative measurements using RT-qPCR are particularly susceptible to inhibition, as small amounts of co-purified inhibitors can have relatively significant effects in downstream analysis. Let's take a look at our current sample of RNA extract below to see how each of the co-purified molecules affect downstream steps involved in RT-qPCR. Co-purified molecules can include proteins, DNA, and other impurities such as residual chemicals left over from the extraction protocol. Proteins and other impurities can decrease the efficiency of two key chemical reactions occurring in RT-qPCR: reverse transcription and PCR amplification. Reverse transcription is the process by which RT-qPCR employs to generate cDNA. You already know from Module 1 that PCR amplification is the process by which cDNA is amplified to quantify the associated fluorescence of a gene target. Therefore, inhibition of either of these reactions will influence the accuracy of detection observed when analyzing RT-qPCR amplitude plots. To show this effect, observe the change in the associated RT-qPCR amplification plots as the current sample is cleared of RT-qPCR inhibitors. Notice that as the sample becomes more purified, the CQ values decrease, showing that the purer the sample of RNA, the more accurate and sensitive the experimental assay becomes. Taking another look at our original RNA extract, you may notice that DNA can also be included in an RNA extract sample. DNA can also negatively affect PCR amplification. With excess DNA, assays may bind to not only the cDNA representing the RNA transcript, but also bind to the DNA that is lingering, causing an artificial inflated signal in the amplification plot downstream. So far, you've seen how co-purified molecules can negatively impact RT-qPCR. However, starting with a purified sample of RNA is not the only factor to consider when attempting successful RT-qPCR. A scientist must also consider the sample integrity as well. Sample integrity refers to whether the RNA transcripts in the sample are intact or degraded. From previous slides, we learned how unstable RNA biotypes are. Thus, it is not uncommon for RNA samples to be somewhat degraded upon extraction. If the RNA sample contains breaks in the transcripts of interest, then both reverse transcription and PCR amplification will likely be inhibited. During reverse transcription, the degraded RNA transcript may not be fully reverse transcribed, which has lasting effects within PCR amplification. For example, the fragmented cDNA will not fully be amplified during PCR amplification, which will reduce the fluorescence signal from the gene target of interest. Let's take a quick look at how RNA integrity affects the CQ values in the amplification plots. Notice how the CQ values begin to increase with RNA degradation, showing much lower fluorescent values than what should be reported with intact RNA. Now that you understand the importance of pure and intact RNA extract when utilizing RT-qPCR, let's consider how these variables may be useful in considering the appropriate extraction kit and sample type, and explore the precautions necessary for your experimental needs. Most often, extraction is carried out using commercially available kits, which have the advantage of being user-friendly, simple, and quick. When thinking ahead to choose the best RNA isolation kit for your need, you may first consider the RNA biotype that you're interested in. The most common extraction method used is with total RNA extraction kits. These isolate messenger RNA, transfer RNA, and ribosomal RNA, but often fail to isolate smaller RNAs. With the explosion of interest in non-coding RNAs like enhancer RNAs and microRNAs, which can vary in length considerably, it is advisable to avoid kits that target all biotypes. It is also advisable to avoid kits that employ targeted column binding or precipitation because these techniques often exclude the very small molecules of interest. Instead, I recommend searching for specialized kits that call attention to your small RNA of interest. In addition to extraction considerations, it is essential that RNA is not contaminated with DNA, since this cannot be distinguished from cDNA in the qPCR reaction, as prefaced earlier. To overcome this, most protocols rely on the use of DNase I treatment, which digests any DNA. When thinking ahead, it is also important to consider the sample type that you may be working with. RNA samples embedded in complex matrices such as FFPE or formalin-fixed, paraffin-embedded samples can often require more specialized cDNA synthesis kits containing engineered enzymes or other supplemented chemicals to account for difficult inhibitors or challenging targets with secondary structures or high GC concentration. To reduce the degradation of RNA samples obtained from tissue and forego the use of specialized extractions kits, opt to use fresh or flash-frozen tissue. Also of note, remember that isolating RNA from tissues will likely yield RNA from several cell types, and different cell types have different RNA transcript levels. To provide some insight into the different concentrations of RNA yields that you may produce from extraction methods, take a glance at the chart and notice the vast differences in predicted RNA yields from liver, spleen, and heart compared with low-producing tissues such as bone and blood. Isolating RNA from cells proves to be the easiest in terms of conserving RNA integrity and decreasing PCR inhibitors. When extracting RNA from cell cultures, check to ensure high viability and attempt to use the same number of cells in the extraction among samples. Before beginning extraction with cells, it is always a good idea to check with the associated kit protocol to ensure that you are starting with the recommended amount of cells. As demonstrated in the previous figure, extracting RNA from blood can prove difficult due to the low numbers of cells contained in blood. You may try concentrating the cells from several pooled samples of blood first before extracting RNA. If you haven't already picked out an extraction kit that's appropriate for your experimental needs, I would encourage you to consider one of the many kits that Bio-Rad has. Listed below, you'll notice that Bio-Rad has a variety of kits designed for several different tissue types, including different types of cells, animal tissues, and plant tissue. The kits are designed to cover a variety of different amounts of starting material, and many contain DNase to eliminate any DNA contamination. In addition to choosing the appropriate RNA extraction kit based on the tissue type and RNA biotype that a scientist is interested in, there are a few precautionary steps that should be performed at the benchtop to ensure a low probability of RNA degradation while preserving the purity of your sample. Let's review a few of these steps below. Step number 1. Always store RNA samples at -80 degrees Celsius. Step 2. Use RNase-free solutions on all surfaces, pipetters, benchtops, glassware, and other laboratory equipment. Step 3. Wear appropriate personal protective equipment, including gloves, lab coat, and safety glasses. Step 4. Avoid freeze-thaw cycles to RNA samples when you can. Step 5. Make sure to implement DNAs, especially if working with samples from organisms with very small to no introns. And finally, step 6. When you can, use aerosol barrier tips. So far, we've explored the importance of isolating a pure and intact collection of RNAs when utilizing RT-qPCR. In doing so, we've reviewed also the importance of pairing the correct RNA isolation kit with the particular tissue type and RNA biotype of interest. Additionally, we have covered some advisable best practices to perform when working with your RNA extract in the lab to ensure the quality of your RNA remains unhindered. Assuming that you have completed a successful RNA isolation protocol, the next step before proceeding to cDNA synthesis is to check your RNA extract to verify not only the quality, but the quantity as well of the extract. After all, it wouldn't do any good to have captured high-quality transcripts that remain too low in abundance to detect, right? Alternatively, it would be just as frustrating to capture an abundance of fragmented transcripts accompanied with several co-purified PCR inhibitors. During these next few slides, we'll uncover common methods to qualify your RNA sample in an effort to validate the quality and quantity before investing time, money, and energy into continuing the remaining steps in RT-qPCR. As we begin to explore these methods, keep in mind that quantity of RNA is largely determined by the yield of ribosomal RNA, or rRNA, whereas the quality of RNA is determined by the purity and degradation status of the RNA molecules. However, there is a significant overlap between the two factors since it is difficult to assess the quality of a sample with low quantity of RNA, and parameters which affect RNA quality may interfere with the quantification process. Let's take a look. Perhaps the most commonly used instrument to assess nucleic acid quality and quantity is the nanodrop instrument. The nanodrop measures the UV absorbance of the various molecules in a sample in a process termed spectrophotometry. Nucleic acids, such as those that comprise DNA and RNA, have unique properties that are observed when measuring their UV absorbance at specific wavelengths, namely 230, 260, and 280 nanometers. If we were to measure each of the UV absorbencies for each nucleic acid at 260 and 280 nanometers, we would uncover a unique ratio for each nucleic acid. If we assume that each DNA or RNA molecule is comprised of an average frequency of nucleotides, we would predict that the string of DNA or RNA molecules present in the sample would approximate the weighted average of the 260 to 280 ratio for the 4 nucleotides present. In fact, this is the principle applied in determining the presence of DNA or RNA in a sample when using the nanodrop. Notice that uracil, the nucleic acid found in RNA, has a higher 260 to 280 nanometer UV absorbance ratio when compared to thymine. Therefore, the average value of RNA will be higher than DNA. When testing a nucleic acid sample using the 260 to 280 ratio, values of RNA should approximate 2, whereas values for DNA should approximate 1.8. This is the general rule of thumb when testing an RNA sample for presence of pure RNA or pure DNA. Thus, the 260 to 280 nanometer ratio can provide some insight into the purity of sample regarding whether DNA or even other molecules such as protein are present. Other ratios, such as the 260 to 230 nanometer ratio and the 260 to 270 nanometer ratio, can be used to determine the presence of organic compounds, sugars, urea, and salts or phenol, respectively. Additionally, the concentration of nucleic acids can be roughly calculated with a nanodrop by implementing the Beer-Lambert law, which predicts a linear correlation between absorbance and concentration. The advantages of using the nanodrop include speed to results, access to equipment, cost, and low requirement of sample input, equating to about 1 to 2 microliters of sample. However, the downside of using nanodrop is that the concentration reported is often a best guess at most due to the heterogeneous mixture of molecules within an extract. The various molecules collected that are not nucleic acids interfere with obtaining a true correlation between absorbance of the nucleic acids, and without an effective method to tease out these effects, the reported concentration is rarely accurate. However, if the reported quality is relatively pure and the reported concentration is abundant, you can likely use the reported concentration to proceed with subsequent RT-qPCR steps. Another commonly used method to assess RNA quality includes gel electrophoresis. Electrophoresis is a method of separating molecules such as nucleic acids based on the net charge, size, and conformation within a matrix. When performing gel electrophoresis with RNA extract, small aliquots of denatured extract are loaded into microwells within a polyacrylamide or agarose gel. The gel is submerged in an electrophoresis chamber with buffer that allows the flow of an electrical current. Nucleic acids have an overall negative charge due to the presence of their phosphate backbone. Therefore, they migrate through the porous gel towards the anode. The size of the RNA molecules determine their rate of migration, in effect, separating the RNA molecules into distinct bands of similarly sized RNA over the course of 30 to 60 minutes. Upon completion of the migratory phase, bands of RNA are stained with aethonium bromide and visualized under UV light. Because ribosomal RNA is vastly abundant within cells, the RNA bands corresponding to this biotype generate a high degree of signal that a scientist can use to interpret the relative presence of RNA from the isolation method used. When examining mammalian RNA, the intensity of the 28S rRNA and the 18S rRNA can be particularly informative. Because 28S rRNA is roughly twice the size of 18S rRNA, a scientist would expect the ratio of intensity of 28S to 18S rRNA to be around 2. Deviation in this intensity ratio is suggestive of RNA degradation. Furthermore, the absence of these bands indicates little to no RNA present. This qualitative method proves to be very cost-effective with a straightforward protocol. However, this method is slower than the nanodrop method and uses far more RNA extract as input. Additionally, electrophoresis proves to be more informative of the quality of RNA, leaving little information about the concentration of RNA. So another method of quantification may be necessary to proceed with subsequent steps in the RT-qPCR protocol. The bioanalyzer is another method to inspect RNA quality and potentially quantity. Similar to the concept explored in the previous slide, the bioanalyzer instrument performs automated electrophoresis to an RNA sample. A proprietary algorithm is utilized in conjunction with the 18S to 28S rRNA ratio to produce an RNA integrity number, or RIN. This value can be used to qualify the degree of RNA degradation in the RNA. A higher value reports a higher quality of intact RNA. Advantages of this system include increased resolution of RNA integrity compared to the gel electrophoresis method mentioned in the previous slide. Additionally, the input volume is minimal, and for high molecular weight RNA, this system can provide the concentration of your extract as well. If you have access to this instrument and you're looking at larger RNA biotypes, this is an acceptable option to validate RNA quantity and quality. The qubit fluorometer represents the last method that we'll cover today in terms of validating RNA isolation. The qubit RNA assay utilizes two unique dyes, one that binds to large, intact, and structured RNA, and the other that selectively binds to small, degraded RNA. Together, they can quickly access the quality and integrity of an RNA sample. Results are presented as a total value for the RNA sample integrity and quality, or RNA IQ number, and as the calculated percent of large and small RNA in the sample. The RNA IQ number is based on a scale of 1 to 10, like other RNA quality scores, wherein a high IQ number indicates most of the sample consists of large and structured RNA. Conversely, a small IQ number indicates the sample comprises mainly of small RNA with limited tertiary structure. If your lab has access to this instrument, I strongly recommend this method as it represents the most accurate and efficient technique to validate your RNA extracts. Now that we've considered a few methods to measure both the quality and quantity of an RNA extract, let's explore how much RNA is required for appropriate detection of a particular gene of interest utilizing RT-qPCR. If we assume that the RNA extract is of high quality and that the polymerase and reverse transcriptase are performing efficiently, and that the quantity of RNA extract is sufficient to satisfy the recommended cDNA synthesis steps within the protocol, then we can begin to make an educated guess of how much RNA extract to use based solely on the abundance of the target. Keep in mind that the process of choosing the correct RNA input for cDNA synthesis can require some optimization. However, as a general rule, the higher the abundance of gene target, the lower the amount of RNA input is necessary, and vice versa. To exemplify this concept, take a look at some recommended RNA extract inputs for the various RNA biotypes. Notice that in an RNA extract sample comprised of all RNA biotypes, the gene target is likely low in frequency, which means that the extract input requirement will be much higher in concentration when compared to an extract sample containing only a fraction of the RNA biotypes within a cell. Although the previous table can be helpful in dialing in a starting input extract concentration, the correct concentration ultimately comes down to trying a few different concentrations and examining your results downstream. Consider the following RT-qPCR experiment. A scientist investigates PKG1 expression utilizing two different RNA extract inputs, one at 100 nanograms per reaction and another at 100 picograms per reaction. The scientist utilizes a one-step process, something that we'll touch on in the future slides, and performs 20 replicates per group. If we examine the RT-qPCR results downstream, we see the following amplification plot. Notice that the replicate CQ values are reproducible and the distance between the CQ values from the two groups of different concentrations approximates 10, which is the expected outcome for 1,000-fold change difference in the target gene. This specific distance confirms that, as we originally assumed, the reactions are performing efficiently. Also notice that the CQ values of either group do not fall within the questionable region of our amplitude plot. With these checks confirmed, we can choose either starting RNA input as a good starting point for similar samples as we continue our experiment. Now that we've examined the processes and considerations involved in RNA isolation, let's return to the basic RT-qPCR protocol to examine the next step. Recall that for this module, we'll be focusing on the first two steps of the RT-qPCR protocol. If we revisit the original experimental example mentioned earlier, we can see that the next step after RNA isolation is cDNA synthesis. Let's continue our discussion focusing on the different ways to incorporate cDNA synthesis in RT-qPCR analysis. There are two methods available for cDNA synthesis by RT-qPCR, the two-step reaction and the one-step reaction. Let's first examine the steps involved in the two-step reaction. As the name denotes, the two-step reaction is split into two separated enzymatic reactions. The first enzymatic reaction is called reverse transcription, and the second reaction is called PCR amplification. As mentioned previously, reverse transcription is the process by which RNA is converted to cDNA. The current figure illustrates the basic steps involved in reverse transcription of mRNA occurring in tube 1. During the reaction, primers bind to complementary regions of the mRNA transcript, initiating the recruitment of reverse transcriptase. Reverse transcriptase utilizes the single-stranded mRNA molecule as template to generate a complementary strand of DNA. When this reaction is completed, a scientist will transfer the newly synthesized cDNA into a separate tube, tube 2, to perform the second enzymatic reaction, PCR amplification. During PCR amplification, primers and potentially probes that are designed to target the gene product or products of interest will bind to complementary regions of cDNA. Polymerase will recognize the bound primers and carry out amplification of the cDNA for typically 30 to 40 cycles. As you learned from Module 1, during extension of the amplicons, if a probe is present, the fluorophore will be hydrolyzed and fluorescent signal will be emitted. Now that we've reviewed the two-step reaction, let's examine the steps involved in the one-step reaction. Similar to the two-step reaction, the one-step reaction includes both reverse transcription and PCR amplification. However, in contrast to the two-step reaction, both enzymatic reactions occur sequentially in one single tube. Choosing the right cDNA synthesis format can be a key step in completing a successful RT-qPCR experiment. To help decide whether the one-step or the two-step reaction is right for your application, let's consider a few pros and cons for each reaction setup, starting with the one-step reaction. Because the one-step reaction includes both enzymatic reactions within the same tube, it is considerably faster when it comes to completing your RT-qPCR protocol. Additionally, the one-step process requires fewer pipetting steps, which can lower the chance of contaminating the sample. However, there are a few drawbacks to consider as well. For example, it's important to note that the cDNA generated from the reverse transcription step in a one-step reaction is not recoverable for storage and continued use for future RT-qPCR analysis or other applications of interest. Additionally, because the reverse transcriptase and polymerase require unique environmental conditions to operate at optimal efficiency, they will likely suffer some suboptimal performance when included in the same conditions within a one-step reaction. In a two-step reaction, a scientist has the option of collecting the cDNA generated from reverse transcription and storing it for later use if desired. Also, if a scientist is interested in targeting a gene product that is especially rare in the RNA extract, a pre-amplification step can be performed with the cDNA yield from a two-step reaction to increase the target frequency before continuing on to the PCR amplification step. Some drawbacks to the two-step process include slower times to results and more pipetting steps, which can increase the chance of contamination within the sample. Now that we've reviewed the two reaction setups that a scientist can perform in the RT-qPCR protocol, let's take a closer look at the reverse transcription reaction itself to examine the enzyme reverse transcriptase. You may have received the impression that all reverse transcription reactions occur with the same enzyme, but this simply is not the case. The truth is there are several different cDNA synthesis kits equipped with several different reverse transcriptases, or RTs. Perhaps the most general and important characteristic that segregates these RTs is whether or not they have nuclease activity, also known as RNase H+ activity. As is shown in the current figure on the right, RTs with RNase H+ activity have the ability to degrade the transcript as it generates the cDNA, thereby eliminating the transcript from being used in any additional RT reactions. The importance of this function is fourfold. Number one, The resulting RT reaction yields a 1-to-1 ratio of transcript to cDNA, a crucial factor when performing accurate gene expression analysis. Number 2, because transcripts undergo degradation upon use as a template, the abundant transcripts are not reused, reducing any chance of biased RT. Number 3, rare transcripts of interest have an increased chance of being reverse transcribed into cDNA. This scenario is possible because as the abundant transcripts interact with RT, they are degraded, which after a short time decreases the competition of transcripts for enzyme and ultimately increases the chance of a rare transcript to contact RT. And 4, the degradation of transcripts will not block polymerase from binding to the potential primed sites on the cDNA as it generates in a one-step reaction. Some common examples of RTs with RNase H+ activity include the avian myeloblastosis virus RT, the Maloney-Murine leukemia virus RT, which is the RT in Bio-Rad's iScript kits, and the human immunodeficiency virus RT. When choosing the right RT for your gene expression experiment, it is important to consider the processivity of the RT, the RNase activity of the RT, and the thermal stability of the RT. As we scan through each of the aforementioned RTs equipped with RNase H+ activity, you may notice that the MMLV achieves great scores for processivity, RNase activity, and thermal stability. For these reasons, this RT is a great pick for most RT-qPCR experiments. Aside from considering the types of RTs available for use in the reverse transcription reaction, there remains to be decided the priming strategy that a scientist chooses as well. The figure displays the steps of the reverse transcription reaction within a two-step RT-qPCR experiment, highlighting the priming strategy implemented within the black outline box. Notice the current priming strategy shown implements an oligo-DT, which binds to the poly-A tail of mRNA transcripts within the RNA extract. However, this is not the only priming strategy employed in RT-qPCR. As shown, random hexamer primers can also be an option to consider instead of oligo-DTs. Random hexamers, as the name denotes, are comprised of random combination of nucleotides that bind to a variety of potential transcripts in regions that reside outside of the poly-A tail. Still, another priming option consists of a gene-specific set of primers. This final priming strategy consists of primers that anneal to the specific gene product of interest. To better understand which priming strategy is appropriate for your two-step RT-qPCR experiment, let's consider the following. Because oligo-DTs bind to the poly-A tail of transcripts, this priming strategy will capture only mRNA biotypes. Due to the position of the poly-A tail on transcripts, oligo-DTs may result in premature termination if the transcript has increased secondary structure. The use of random hexamers as a priming strategy can allow for increased transcript coverage, but may lead to an overestimation of copy number due to an internal priming or a 3' to 5' bias. When it comes to analyzing gene expression, gene-specific primers is the strategy that will suit your needs. For all other applications, I recommend a mixture of oligo-DTs and random hexamers.